Bismillahirrahmanirrahim. I'm Dr. Tahira Bashir, and today's lecture is about the different types of transcriptional factors, how these transcriptional factors they are responsible for the initiation of a transcriptional process. Cis-acting elements, these are basically the nucleotide sequence which are present on each gene so as to regulate the expression of each gene. These elements they lie on the same DNA on which gene is present. So these elements they are known as cis-acting elements. Here are two sets of cis-acting elements are present for both genes. Each gene carry their own elements. Elements mean the nucleotide sequence. This one is the gene 1 from which it contains the set of regulatory elements, regulatory nucleotide sequence which are present on the same DNA on which this gene is present. Similarly here the gene 2, it also contains a set of regulatory elements. These regulatory elements they are differ to that gene, and these two different sets they regulate the two different genes. By means of these regulatory cis-acting elements, each gene it is regulated and also expressed. And other factors which are known as the trans-acting factors. These trans-acting factors are so called because they are proteinaceous in nature. They are transcribed by certain genes. After their formation, their messenger RNA go to the cytosol from where translation take place. Post-translational modification also take place here, and after then these mature proteins then go to the nucleus so as to transcribe another set of genes. So by their transient formation as well as the movement, they are known as the trans-acting factors. Many types of trans-acting factors are present, maybe a general transcriptional factors or maybe a specific transcriptional factors. General transcription factors, they recognize their promoter region, and these general transcription factors they recruited to their one by one. A serial recruitment of these general transcription take place. Serial recruitment of these general transcriptional factors take place so as to activate the RNA polymerase 2, which is responsible for the synthesis of messenger RNA of a specific protein. By the recruitment of these transcriptional factors, general transcriptional factors and other factors, they initiate the transcription. One of the example of general transcription factor is TF2D, transcriptional factor 2D. This transcriptional factor it recognizes its specific promoter region and bind to the Tata box. This is specified for the Tata box. After the binding of the Tata binding protein, then it initiates the general transcription factor TF2D, which then bind to the Tata binding protein. And also it is responsible for the recruitment of another factor like TF2F, TF2H. The TF2F factor it also responsible for bringing the polymerase. As polymerase or holoenzyme, it lack the recognition of its specific promoter region, so it required a general transcriptional factor TF2F mostly in most of the cases in eukaryotes. Then after its recruitment, another transcriptional factor TF2H, it has the helicase activity, also the kinase activity. It actively phosphorylates the polymerase. After phosphorylation of the polymerase, it escapes from the promoter region and now the transcription enter into the elongation phase. The specific transcription factors they are also required to mediate the frequency of initiation and also mediate the responses of different response agents. these specific transcriptional factors, they bind to their specific regions, which may close to the core promoter region. Core promoter region is specified for the general transcription factors, or may thousands of base pair away from the core promoter region. Promoter region may be a core promoter region, may be proximal promoter region, or may be distal promoter region. Here the binding of the different factors to their different regions. For example, GC box, it is responsible for the binding of SP1 transcriptional factors. CART box responsible for the binding of the CTF factor. Similarly, Tata box binding for the TF2D transcriptional factors, and then the polymerase 2 also bind these transcriptional factors so as to mediate the transcription. These are the different regions. Each transcription factor, they bind to their specific nucleotide sequence. For these, they contain the specified motifs by which they bind to the DNA. So each transcription factors bind to their specific region so as to mediate the transcriptional process. The Tata box, which bind the Tata binding protein in some of the gene, but mostly the genes contain initiator. Here the initiator box, specific nucleotide elements, and similarly the DPE, downstream promoter elements. Certain genes, they contain the Tata box, but most of the genes, they contain the initiator or downstream promoter element instead of Tata box. These are the core promoter region, but close to the core promoter region there are the proximal elements or the proximal promoter region, which can bind the GC or CART box. It comprises maybe a GC box or maybe a CART box. And other regulatory elements mean the nucleotide sequence, which are regulated, which regulate the gene transcription. The other elements, they may be enhancers which can promote the enhancer, and after binding with the activators they can promote the transcriptional process. Or the repressor elements. Repressor mean these are also the nucleotide sequence. Whenever they bind with the specific proteins which are specified for their repressor elements, they may repress the gene transcription. So enhancers and repressor elements, they are present. Thousands of base pair from the transcription start site. Thousands of base pair from the transcriptional start site. The basal expression of a gene, they required the recruitment of the proteins on the core promoter region, also on the proximal promoter region. But for the gene regulation or the mediation or the gene expression, to mediate the gene expression, there also required the recruitment of the specific transcriptional factors which on the enhancers or repressors. Enhancer bind the activators, and by means of mediators they interact with the general transcriptional factors along with the transcriptional factors which bind on the proximal element so as to mediate the gene transcription. By means of this interaction, a gene can be transcribed in a speedy way. These are also the different transcriptional factors which bind on their specified region. Tata box, which bind the Tata binding protein, TF2D TF2F, TF2A, TF2B, H, E. All of these general transcriptional factors, they are recruited in a series. The recruitment of one general transcription, it will initiate the recruitment of other transcriptional factors so as to form a transcriptional complex. This one is the formation of a transcriptional complex, and this complex, it will initiate RNA polymerase 2 so as to start the transcription from the transcriptional start size. However, these general transcriptional factors, they also functional when they interact with the specific transcriptional factors or other factors which bind on the proximal promoter region or on the enhancers. There are the serial recruitment of all of these transcriptional factors on their specific region, on their specific nucleotide sequence, or on their specific promoter region. Recruitment of, first of all, the Tata binding protein on the Tata box, then it will recruit the TF2D, another transcriptional factor, then recruited the 2A and 2B and other transcription factors, then the recruitment of 2F along with the RNA polymerase 2. RNA polymerase 2, it is not recognized alone. The holoenzyme in eukaryotes, the holoenzyme of RNA polymerase, it is not recognized along the promoter region, but it requires certain transcriptional factors. Interaction of these transcriptional factor with that of the RNA polymerase 2 will initiate the transcriptional process in eukaryotes. So the serial recruitment of these general transcriptional factor after the recruitment of the specific transcription factor on their specific region and the recruitment of these general transcription factors along with the interaction, all of these processes, they are needed to form a mature transcriptional complex so as to start the transcriptional process. This is the this picture shows how the specific transcription factors which bind to the enhancer region and the general transcription factors and the factors which bind on the proximal promoter region or the general transcription factor bind on the core promoter region, how they interact with each other. One hypothesis is that they only interact each other so as to form a functional transcription complex. There is need of a bending of a DNA molecule because enhancer elements, they are thousands of base pair away from the transcriptional start site so as to form interaction between these transcription factors along with these specific transcriptional factors or protein-protein interaction. There must be bending of this long loop of DNA, and this bending held by the DNA bending proteins which stabilize this bending and also promote this interaction so as to form a mature transcriptional complex along with the polymerase 2. When enhancer bind the specific transcriptional factor and along with the recruitment of the general transcription factors on the Tata box with polymerase 2 and mediators, how they form a mature transcriptional complex by two ways. In first way, they are recruited one by one like TF2D, recruitment of TF2D, then 2A, then B, E, along with the polymerase H, F mediators, then form a mature transcription complex. In other way, these enzymes, these are transcriptional factors. They are first of all they form a complex, and then this complex, it will recruited to their specific region or specific nucleotide sequence on a DNA so as to mediate the transcription. So by two ways, maybe this one or maybe this one, different transcription factors, this different proteins, transcription factors are actually the proteins. They are recruited to their specific places so as to form a mature complex. Enhancers, they are basically the nucleotide sequence which are present thousands of base pair away from the transcription start site, but it is not necessary that they always present upstream of the transcription start site. They may present upstream or downstream from the transcription start site, or it is also it may be present close to the transcription start site, may be present thousands of base pair away from the core promoter region on which the general transcription factors recruited, or may they close to the promoter. So enhancers, they are basically the response elements. Elements mean the nucleotide sequence. Enhancers, they are the response elements. When hormone bind to their specific enhancers, then they mediate their responses by the recruitment of the specific transcriptional factors on their specific promoter region so as to mediate the transcription of a specific gene. These are by this way, an enhancer when bind to a response agents, they can mediate the transcription of a specific gene. Enhancers may be upstream from a specific gene, or these enhancers may be downstream from a specific gene. Here the different transcriptional factors, how they can interact. For gene A, these set of transcription factors required, and gene B are different set of transcription factors when bind to their specific region take place. DNA bending protein, they bend the loop of DNA and they are responsible for the interaction of the specific transcriptional factors which are present on the enhancer or bind on the enhancer to the general transcription factors through mediators. Here the different mediators, different set of specific transcription factors and the general transcription factors, they are required for a gene B, for the transcription of a gene B.